no problem. Okay. I've been trying to speak Spanish in the weekend, and it's challenging. So second languages can be tough. So it's no problem to go slow. And so the first, we'll start with introduction to the course, introduction to us. We'll have you introduce yourselves, tell you a bit what we're going to do, and then we'll walk through kind of what the course is going to be. The course has three sorts of lessons types. One is lectures, where we'll just talk, and you can ask questions, please, in any time. Put your hand up, it's okay. And the second is computer part, where we'll be working in R and on the command line in Unix to do analyses. That's what the sticky notes are for. We'll explain, but so, what is this? Sign yes? Science classes. Ah, okay. I can't be in trouble. Yeah.
Se ha hecho pesado, No, pero que me vas a ver.
No, pero quería libre, pero no puedo porque es más difícil
Ah, ya.
Ojalá pueda asistir en las tardes, pero no sé si quedará profesor así.
Translate the Spanish for us. Speaks good English. Yeah.
Speak English?
Yeah, can someone just try to help us translate?
Yeah, okay.
Gracias.
Hay un classroom.
There's a classroom in Google.
Ya, deben subir los papers.
You need to upload the papers,
Calificaciones.
the grades,
Okay. Yeah.
Asistencias.
assistant the list.
Okay.
Las claves les han enviado a su correo.
You have the password and your payments.
I don't think we got a password. No.
Okay.
Ya,
No
ok.
password.
Les voy a reenviar el correo.
She's sending.
¿Ok?
Okay. Gracias. Yeah, we. Okay. That's
Último.
Okay.
No firmitas.
Gracias.
Gracias.
Yes, you tried to get him to the classroom last night, didn't you?
Yeah.
Un delegado, alguien para que
firme las asistencias y me las entregue al final.
Fernando.
Ya.
That's okay.
Son demasiados documentos. Universidad Nacional.
Those are yeah, we haven't set up the Google Classroom yet because we didn't get a password yet, and we also we don't have internet access yet, so we are a little bit limited, but we're prepared. So the Google Classroom I think will be online, I hope tomorrow.
Yeah.
But we need to get the password from Karen and things, so we'll do our best. Okay, so as I was saying, the second type of sort of module in the course will be programming. That's what these are for. I'll explain. You don't have them, probably everybody else does. I'll give you some info when we need to. And then the third type will go to the lab, or his biodiversity lab across the way, and that's where. And I will lead the programming lectures, and Robin will lead the lab components across the street, and we'll do the entire DNA barcoding laboratory workflow. What I say, that's okay. It's okay. Yeah, sit anywhere. But one of you has already prepared some samples. So which one of you prepared the samples?
DNA.
It was you, Lydia.
Oh, it was you.
Okay. Okay. So ichneumonids, wasps.
Yes.
Yeah. So we'll Barconids. Okay. So we'll do some DNA barcoding of Barconid wasps. But we've already got the sample part, so we'll just go from the DNA to the bioinformatics. See?
A few samples.
A few samples. Okay. Any questions so far? Okay.
Will there
Are you-
homework?
Homework? Do you want homework? No, that's okay. No homework. I'd say.
Only if any background reading, like say we mention something and we don't explain very much about it, maybe we'll just tell you, okay, Google read something more about that in your own time. But no homework other than that, I don't think.
If any of you want to do more work, optional work, there are some additional things we can do. So on Thursday afternoon, if anybody wants to do a metabar coding laboratory workflow, we can do that because there's no class scheduled Thursday afternoon. So that's optional. And if anybody finds the computer programming too easy, I can give you harder things to work on. So optional homework, but not necessary. Okay, so we will start just going over who we are and our backgrounds, and then just to meet all of you, have you just tell us a little bit about what you're working on, what you do, talk a bit about what we're doing in the course, and then we'll do a sort of more traditional lecture on DNA barcoding. Okay, so I'm Ken Thompson. I did my PhD, finished in 2021 at the University of British Columbia in Vancouver, Canada. So on the west coast. Just for context, Canada is a very big country, and the distance between my hometown and the University of British Columbia is bigger than the whole country of Peru, top to bottom. So very big. I worked with Dr. Dolph Schluter, who's an evolutionary biologist. I did speciation, so new species formation, genetics, and I worked on little fish. And then I did from 2021 to last year, I worked in the United States, Estados Unidos, at Stanford University with Molly Schumer, where I did more genomics, whole genome analyses of fish and plants in the desert. And since last year, in September, I work at the Center for Biodiversity Genomics in Guelph with Robin, where I do research and scientist and project managing. And just a bit more. So I work on metabar coding research and development with the innovation team with Robin at the Center for Biodiversity Genomics. And then I coordinate projects where we're working with mines and other companies on using DNA barcoding and metabarcoding. Hola, you can get it. Yeah, hey, buddy. Yeah, come. There's this. Fun fact, just for fun, I play drums, baterias, in a band, and I have dos gatos, 2 cats. I learned some Spanish at Kennedy Park in Lima. The gatito, right? Gatito, nombre Jifuri, gata, Junimo. So those gatos. Okay.
Hi, yeah, on to me. My name's Robin. I'm originally from the UK, Scotland specifically. I don't know how far you can tell, like accents in English, maybe you can tell my accent is a little bit different from Ken's, because he's Canadian. But I'm a dual citizen, I'm Canadian as well. And I did my PhD a long time ago, that was at the University of Edinburgh, back in 2003. My PhD supervisor was Mark Blackster. And my topic was soil nematodes, so I was doing kind of an early version of DNA barcoding using DNA to see how many different kinds, different taxa of nematode worms are living in soil. So they're free living ones, not parasitic nematodes. And I worked after that, immediately after my PhD, I worked with the British Antarctic Survey, so I was doing similar things on marine nematodes, and I did go on a research ship down quite close to Antarctica for a few months, so that was quite exciting. And then I worked at the University of Guelph. I did a postdoc with Paul Bear originally. But then I kind of moved away from the biodiversity world. I worked in a lot of different areas. I'm kind of a general molecular biologist. That's really my skill set. Moved back to the UK for a period of time. I worked in Cambridge University at the Stem Cell Institute, so kind of a bit more medically focused. But then came back to Canada just a couple of years ago due to my wife getting a faculty position actually at the York University in Toronto. And now I work at the Center for Biodiversity Genomics, and I'm the co-lead of the innovation unit. So it's me and another colleague called Sean. We have it's the Small Innovation Lab, and it's basically the methods development lab for the Center for Biodiversity Genomics. CBG is really like a big DNA sequencing factory. It employs, I think, 100 or 150 people. Does sequencing on really a big scale. But our lab is sort of a smaller group that does kind of smaller, more experimental projects and trying to find sort of new ways of doing sequencing. So my focus is kind of methods development, optimizing protocols for barcoding and metabarcoding. The main kind of the sort of direction of our research, if you like, is what we're calling distributed barcoding. That is finding ways to make sequencing protocols easier and cheaper and more accessible so that smaller labs and labs in lower income countries can do DNA sequencing themselves rather than sending their DNA to us to sequence. And so DNA barcoding becomes a thing people really can do all over the world. And fun facts about me, I've got 2 children, one of them are at age 6 and age 9, and I chose this picture in particular because there's a thing called snow, which I don't know how familiar you are with in Peru. I guess in the mountains you get snow in Peru, but not in this area. Here I don't think. Yeah, Canada we get a lot of snow, at least in winter time. The fun fact about me is that I collect this series of Japanese robot toys called Zoids, and that's one of them there. A big collection of those. It's my hobby.
In fact, I didn't know that about Robin until I saw.
Yeah, we were just talking. Okay, give me a fun fact to put in the talk, and I thought, okay, well, I'll put that in. It's just
is this visible? Or I wonder if should we turn off this light maybe? Let's see. Who here has walked on snow? Put your hand up if you have walked on snow. No one? But you have one person. Yeah, too. Cool. If you come to Canada the right time of year.
Yeah. Well,
Is that better?
That's better.
It's
No
better.
one will fall asleep? No? Okay.
Yeah, because there's still. That's it.
Okay, so I'll just tell you a bit about the institution that we work at, very briefly. So the Center for Biodiversity Genomics, primarily most people work on, it's a biodiversity factory, kind of like Robin said. There's 20 to 30 people that all they do is they take insects or other samples and they put them into wells in plate, or they pin them. Then it goes to the big genomics core facility, goes to imaging lab, and it's really just a big barcoding workflow. And the goal this year is to do 3 million DNA barcodes. So that's 3 million specimens where we have the actual specimen, DNA extracted in the museum, in our freezer, image, and a DNA sequence online. And then the International Barcode of Life, which is sort of the other sort of organization that we're a part of. It's a not-for-profit organization that really works with the global research community to steer the global and coordinate big barcoding projects. Has big conferences every 2 years. So Robin and I have been to South America 3 times in the last year. First time was in Brazil to go to the conference. And then next year we will go to Thailand for the conference. So if any of you do barcoding, there's usually opportunities for students to go, for example, to the conference. So keep an eye out for that. And this is the facility. So who's doing exchange at Gulf here? So Valia, you are. Manuel.
Lydia was there.
Well,
Right. So you'll all, you'll work here probably. And so, yeah, that's nice. Okay, and so just brief, just so we can know you. Can we all sort of say your name, what you're doing, how much experience you have with barcoding, metabarcoding concepts, molecular biology, so pipetting, sequencing, hands-on, and then computer coding. And then if you want, no pressure, fun fact or hobby, you can start at the end.
My name is Lydia. I'm interested to parasitoid wolves called micrograss training. I don't have experience to do barcoding DNA, sorry for that, but I want to learn about that.
Yeah.
So I focus to understand that in the moment.
Okay. Okay.
What this course is for.
Yeah. You don't need experience. The idea of this course is, if you have no experience, it's okay. It's no problem. So yeah. Okay. Any fun fact? No, it's okay.
No. It's not.
Okay. Okay.
Hello, my name is Noemi. I am biologist. My thesis, we investigate marine fish, Anisotermus scapularis or Peruvian ram.
Okay.
I investigate nutrition of this species,
Okay.
of cheetah.
Yeah.
So,
Okay. And experience?
no experience.
Okay. No problem. Okay.
yes. My name is Manuel Saucedo. My researching is in stainless steel. I have a little experiences with DNA barcoding, metabarcoding also. I start in metabarcoding with microbiota of plants.
Okay.
Right now is in insects, gut microbiota. The fun facts, I like drought. I like ride bicicleta, cycle.
Cycling. Okay.
Bike.
Biking.
Yes.
Bike.
Cool.
Is all.
Is that it?
Okay.
Okay. Fernando.
Yes, my name is Fernando Messias. I am veterinary,
Okay.
and I'm working now with comparative genomic analysis of hormones,
Okay.
which affects fishes of aquaculture. I am looking for virulence and antimicrobial resistant genes, but I'm here for the group to learn about metabarcoding because I want to apply this to fish
Okay.
microbiota from instantly gills, skins in the future.
Okay.
I only know something about theory.
Okay. But you do computer programming?
A little.
Okay. What is your name?
My name is Mariela.
Maria.
I'm Marilyn. My research is in protection and molecular in small.
Okay. And experience? Little?
No.
Okay.
thesis about secondary ancient DNA with some difficult extract because DNA a little erratic. Fun fact about me, I like cats and dogs. I have two pets.
Okay.
And I love animals.
Okay. Great. Me too.
My name is Andre. Oh, my name is Andre. I'm biology, especially ecology. And my investigation is about what we think sex with stressors. I don't have experience in metabar coding, but I have some theoretical basis.
Okay.
Fun facts, maybe I collect Star Wars figures.
Star Wars
Star Wars
figures.
figure. Okay, so you're a toy collector as
Especially
well.
vintage collection.
Okay. Very good.
Very good.
Okay.
Hi, my name is Alexandra.
Thanks.
Well, I investigate about plants, especially orchids. My thesis is about barcoding in orchids. I really want to know about more engagement, and this is the best one. For getting further precision to the thesis in barcoding. My experience, I have a little more of the theory,
Okay.
but I really have more practice about this.
Okay.
Well, I love sleep.
You love what?
Sleep.
Sleep. Okay. Door. Great. Me too. Especially after a long flight, sleeping is really
Oh yes. I just arrived in Lima yesterday, and that was an overnight flight. I didn't sleep.
Okay.
My name is Stephanie. I have a bachelor's degree in genetics and technology, and I'm now in the master programs of bioinformatics and food science. I'm researching. My thesis in master is about using airborne and environmental DNA.
Okay.
Combined with nanoproduct sampling to evaluate plant biodiversity in Corrientes area, an important area in Peru. And about my experience with metabar coding, I have a little with my undergraduate thesis, and it was about to evaluate the interaction about microbiome and soil microbiome and metabar. So I have experience about metabar coding on my own and also with the help of bioinformatician.
Okay.
And fun fact about I have a cat and a dog.
A cat and a dog. Okay. I noticed in Peru, all the dogs have coats. It's funny. It's good. Okay.
Hello, my name is Cynthia Gallardo. I am a biologist and microbiologist and parasitologist. And my thesis is about bioorganization using native bacteria of the soil, also the native soil of environment. Well, this will be my first experience with metabar coding methodology. Fun fact, I have 3 children, so I don't have one, but usually play with them.
Yeah. Good. Okay.
My name is I am environmental engineer. My thesis is about microbiota in soils, tissues, and foods of cover. It will be analyzed by metabar coding. I have some experience in biocoding and metagenomics.
am Pablo Sanchez. I am an entomologist working on systematics of cover flies, which are predator flies. Yes, absolutely. I have no experience with part coding or computer coding. I work mostly with morphological data.
People say both ways.
Okay. Fun fact, I like soccer. My team is the Korean Champions.
Okay. I know Jorge's Crystal?
No,
Crystal? Jorge's team? Is it Crystals or something?
Cusco?
Crystal?
Cristal.
Cristal.
Okay. You want to skip? Or you?
One year ago, I did an analysis in guinea pigs. Guinea pigs, yes.
Okay. Cool. You like red hot chili peppers? And sauerkraut.
My name is Ena. I am a biologist too, and my project is about parts of plankton. And I have some experience in DNA barcoding, but in this project I try to implement the nanopore sequencing. So this is
That's exactly what we're doing in this course.
yes,
So,
exactly.
yeah. Have you done a bit already? Do you have some experience with that? Oh, you haven't done it yet. Okay, well, this will be very useful.
Well, the fun part, I was prepared our sample for the colleagues.
Okay. Right. Gracias. So we'll mark that.
We'll be using your samples then.
Okay. That's okay. Great. Oh, this turned off. I think it just went to my computer. Thanks, everyone. Very nice to meet you all. It's good to hear about the range of experience. The students with more programming experience, it'll be your job to help the other students too. So we will do our best to help, but it's a collaborative teaching. We'll use your help. And so I can explain now. So I gave most of you these. Who doesn't have? Put your hand up. Okay. So just take one of each. The yellow, amarillo, means you're working. So this is when I say, let's do some practice on the coding. Amarillo means you're working on it. If you're stuck, you can't proceed, you change to pink, rosada. And then me or somebody else, will try to help. And then you're done, you take a verde, and you put them on the computer, back of your computer monitor, so that way I can see. And then I can see most people are done, most people have completed, and then we can move on. So when you start coding, Amarillo, stick here, and then switch as you progress. Everybody else has? Okay. Okay, so in the course, here's what I want you to, we want you to finish with. So you understand really the core concepts about barcoding and metabar coding, what each is, why you would do certain approaches, what it can versus can't do. Learn really the main important protocols for the modern approaches for barcoding in the molecular lab. And so Robin will be the person, so you will learn what we do, right? We're developing new methods all the time, and we'll teach you those. So you'll be really on the 2025 current techniques. Get some bioinformatics skills of how you analyze data that you get from the sequencer, and do analysis of the data. So those are the main things that we'll try to learn. During the course, ask questions if something is confusing. That's no problem. That's what we're here to do, answer your questions. And I said this before, if we're talking, Robin and I will both try to speak more slowly. But if we're talking too quickly, you can just ask us. We won't be offended. You can ask us to slow down. I speak a little French, and all the time I ask people to slow down when they talk so I can understand. So it's okay. When we're coding, what you'll do is, when you're working on it, stick sort of here. Just working on it, and then you switch. So that's how it goes. That way we can see. So I couldn't find those exact colors. Although I guess I had orange. But working on it, stuck, I'm done. Okay? For the programming, the computers, as I understand, have Ubuntu and Windows installed. I would recommend using the Linux or the Ubuntu on the computer when you boot them up. I don't use Windows. I haven't ever really used Windows, so I don't have very much experience with it. You can use some. Have any of you used like Windows subsystem for Linux? Anyone? No? Okay. So you can use Windows to do these types of analyses, but bioinformatics analyses generally are being done on servers and clusters which use Unix. And Ubuntu and Mac are native Unix or Unix-like systems, so it's better. And we'll install some packages in R. We'll use R and RStudio. Who's used RStudio? You put your hand up. RStudio. Half, maybe?
More or less.
Okay, that's good. So we'll use RStudio. Who's used an R project in RStudio? One? Our hand up, R project? Just two? Okay. So we'll use something called an R project, which is a really nice way to work in RStudio. And by the way, that's good. I was hoping that building the course, the difficulty, I didn't want to make it too hard or make it too easy. So I was expecting most of you to have not used, many of these things. So this is good. And you need to access the terminal or the command line, and we'll go slow through this. But on the Unix system that you have here, can you open like a terminal window or a command line? Is it doable? So yeah, that's good there. Yeah. Can you type? Noemi, can you type ls and push enter? Yeah, good. Okay. So how did you do? What did you click for that? How did you open it? And then is it just here on the side? What's it called? If you go to the app. Terminal?
Terminal.
Okay. So make sure you can open that, and if you can't, somebody else can help. So that's good. Okay, and this is new for now, but really the key for coding is to be able to use and troubleshoot software that other people have written. That's really what most coding is now. You're not going to be developing a lot of long pipelines yourself anymore. That's, especially for barcoding and metabar coding, it's very standardized. So you kind of just have to understand your own applications and how to use the software. And maybe 10 years ago, 5 years ago, I would usually read discussion boards and forums online, Stack Overflow, if you know this. But now, almost everybody uses AI to help code, and that's good. You should do it because it makes it faster and you can learn. But it's still important to understand the basics and how to do things yourself. So when you're doing this course, I will sometimes ask you to do challenging things, and you should use ChatGPT or similar, because that's what everybody does, and it'll make you better. You don't want to, if you don't have internet, you don't want to be able to do nothing. So you need to learn some basics, but this is okay. So not cheating, it's good, okay? Yeah. But sometimes, when you're doing biology and research, ChatGPT can't help so much. So you need to still have some skills, because when you're doing research, you're usually the one developing the knowledge that eventually ChatGPT will use. Okay, so here's what we can do, and I'll put this on the classroom when we get that tomorrow. So today, we'll have another lecture on barcoding. We'll do computer lab, introduction to R Studio, and basic command line in Unix. And then we'll go over to the other lab, and Robin will he'll do a little lecture here on some background, and then he'll show you pipetting, DNA extraction, and PCR as a demonstration.
Well, we're not actually doing DNA extraction because in this case we have the samples extracted already. Right. So I'll talk about that a bit, but we're not going to actually do that. We'll just do extraction, pipetting, and PCR.
Right. Thanks. And then in the afternoon, Robin will give a lecture on DNA sequencing and library preparation concepts, and then also with nanopore. Then, Robin, you want to say what your wet lab will do?
Yeah, so we're going to, first of all, check on a gel, the PCR we ran in the morning. And then for the practical, there's going to be, we have, I think, 3 plates of DNA extracts to work with. So we will set up 3 PCRs in total. And the PCR is actually going to be really easy because we had brought pre-made PCR plates with us, which I'll explain a little bit how we've done that, if that sounds confusing. And then, that will be the basis of what we're going to sequence later in the week, assuming.
And then after that, we'll come back here, and we'll have lunch as well between those. So almuerzo and almuerzo, right? Then we'll do at the end of the day computer lab, a little bit more intermediate data science in R using tidyverse and dplyr, if you know this. So it's very good modern skills to learn. And again, I'm trying to make it not too easy, but also not too hard. But if some of you are really, really advanced, you might find this a little bit easy. And talk to me, and we can work on more difficult things. Or if it's too hard, we can help too. But try and make it just right. Tomorrow, we'll talk a little bit more about the bioinformatic approaches we are using now for DNA barcoding. We'll introduce some software that the team that Robin and I are working on have been developing, along with a couple of our colleagues, called Ontology, which is what we will use to do the analysis of the samples for this course. And this is just software that makes the bioinformatics very straightforward, where you can just click a button. Do any of you have Mac computers, your personal Mac computers? We have a couple extra. So I have two extra Mac computers that we can install, and you can work in groups to use Ontology. And then I will use the software, and I'll walk you through how to do it. And this is software that eventually will be free for all of you to download and use.
It's a work in progress. Yeah. It's not totally finished. It's not something you can take to use, but we will demonstrate. But we hope very soon it will be something we'll make public for.
Yeah. So we've been working on this for, I guess it's probably been a couple of years that Robin and his team have been working on it, and I've been involved for about a year. So we'll use an alpha version of the software. So really, you'll be some of the first people to use the software that we hope everyone in the world eventually will use. So, which is cool. And then in the afternoon, we'll be tomorrow in the wet lab over across the way, the molecular lab, and Robin will do DNA sequencing demonstration practice.
So this is how you actually do library prep for Oxford Nanopore, and hopefully, if everything goes well, there'll be enough time to get an actual Nanopore run going by the end of tomorrow. So we will then have some results of that to see the next day.
Wednesday, we will talk about metabar coding and talk a little bit about some of the work that our team has been doing to develop bioinformatic pipelines for analysis, which we're calling Maple. We won't run the full pipeline because the computers are not quite powerful, but we'll run parts of it. We'll do little things on example data. Then we'll do one level up of data wrangling and analysis in R, so make things do a little bit more advanced functions and data science tricks and tools. And I took a course like this when I was doing my PhD, very similar to this course you're taking. And after the course, the way I did my own work completely was different because I learned some of these basics. So I'm hoping that by Friday, the way you do your own computer work is different and you learn a lot. We'll do some. I'll also just do some metabar coding analyses on the command line. The Miriam, the IT person, I'm not sure if she was able to install the software we need, so I might. We'll see how it goes. We'll do our best. And then we'll go over to the Jorge's lab, and we'll do a PCR exam. And Robert, do you want to say what that will be?
Yeah, this is, well, plans are still somewhat loose here. This is the first time we've ever given this course. I'm not sure exactly what the lab setup would be. And there are quite a lot of you here, and the lab isn't that big, but my thought is that we just have each of you do some practice with pipetting, maybe like a row or a couple of rows out of these test DNA plates that we have, run a PCR, and then after that we'll run a gel from each of us and just confirm that each of you can get PCR results, and we'll consider that part of your assessment.
Yeah, and maybe we'll program a thermocycler or something.
Yeah, maybe we can do inputting data into the thermocycler.
And I should say, all of these slides, they'll be on the classroom for you to download, so you don't have to, you can take photos if you want. Still waiting on the classroom. So Wednesday afternoon, we'll do sequence alignment in R and use a new R package to work with the barcode database, BOLD, called BOLD Connect R. Then we'll go, and Robin, you can say a bit about
yeah, this is just another part of the library prep process, which you will see me do this when we go through the library prep. But this is purifying and extracting DNA with magnetic beads. So we should have enough of those beads and enough sort of leftover PCR products from the things we have that we can give each of you a chance to do that process. It needs tubes, it needs magnetic racks. I need to find out from Jorge exactly how many magnetic racks we have, because that can be difficult for a lot of people to do. So again, plans are slightly up in the air, but yeah, that's hopefully what we're trying to do. Just out of interest, has anyone done MagBead cleanup before? No, probably not that. Okay, so that will be to you guys.
It's very cool. It's also the step, when I do DNA work, it's the step I make mistakes on. So it's good to learn.
But it's a very commonly used method in any kind of library prep, any sequencing platform or Scramble or others. Pretty much all of them use some version of this protocol, so it's a very useful thing to learn.
Yeah. And next Monday, when we ask, have any of you done bead cleanup? All of you will say yes. So that's good. Okay, we'll do another computer lab on Wednesday afternoon. That's more applied data analysis. So that will be taking the skills that we learned from Monday, Tuesday, and Wednesday morning, and actually doing it with sort of barcode data and doing some, biological analysis of the data, and a little bit of statistics. I'm assuming most of you have taken statistics course at some point in your bachelor's degrees, so we'll do very basic statistics, and then we'll talk about the final project that we'll do on Wednesday afternoon, and then Thursday morning, we will use Ontology our software to analyze the sequence data that comes off the sequencer, assuming that works. If not, we'll use it to analyze other data that we already have. Then you'll work on your own projects, where I have sort of suggestions for projects, or you can do a different project if you want, and you'll work in groups, 3 to 4 people, to analyze data in response to a specific question. Then you'll write a very short report, 1 page maybe, 1 figure, 2 figures, showing your analysis. And also you'll make a very short presentation, 1-2 slides, and you'll present to the class what you found. And then afternoon on Thursday, there's no class, but if you want, we can work with you more in the lab to, probably with fewer people, more one-on-one, to practice the library preparations for metabar coding, which are the same for barcoding with, it's easier, right?
It really, the library protocol itself really looks the same regardless of what the DNA actually is. So it won't, from that point of view, it won't be anything you haven't seen before. But maybe if we go through it again, maybe that's a chance for some of you to just get in and I'll say, okay, you try this pipetting bits and get some hands-on experience first. The first one would just be you guys watching me do it.
Yeah. So if you're doing metabar coding in your research and you want more experience and hands-on, then Thursday. But let us know, talk to us if we can talk on Wednesday or Thursday, but let us know if you want to do it. That way we know to plan for it. Friday, you can prepare your presentation. You can you'll give the presentation and then that'll be the end of the course. Yeah. Okay, so we have to give a grade for the course because it's an official university course. So 25% is discussions in the class, asking questions and participating in the computer lab. 25% is, participation in the molecular lab across the way. And then 50% is final report, presentation, and also your code for the analysis that you write, you'll save that and send it, and I'll make it. But I should say, don't worry about the grade. As long as you are engaged and doing the basics, you'll do well. Okay, so it's nothing to worry about. Just participate, be normal, and you'll do well. Okay, so no stress. It's all good. Just learn. Okay, you'll do well. And so, yeah, just a note on the difficulty of the course. So, talking to Jorge, coming from where we are in Guelph, it's not easy to understand exactly the background of the students. And after this course, I'm going to Ghana and West Africa and teaching the same course, and the students there don't even speak English. So it's going to be it's a lot of variation in different things. So we expect a lot of variation. If you're very experienced, this might be easy, but I've tried to put more challenging things for you to do in the course. But if it's too difficult or it's too easy, we can make adjustments. So, yeah. Okay, so that's the first one. So we can move on now to the first lecture here. Yeah. Pretty good for time. Yeah. And we'll take breaks too. We'll take, this is not going to be that long. We'll take a little break. You can go walk around, go to the baños, whatever. So far, any questions about just the course structure? Is good? Homework optional? Nothing. Don't stress about the grade. Just focus on learning. Nothing else. Okay. So now we'll have a introduction to barcoding and a little bit of background. So we'll just do this, then we have introduction computer lab, then we go across the way. Okay, so we'll talk background, methods, why we do barcoding, and then some of the challenges we have. So barcoding as a field of study began in 2003 at the University of Guelph, and the basic idea is that you use a very small bit of DNA, and you can use that to identify what organism that came from. And because you use the same part of the genome, part of the mitochondrial genome, for everything, you can compare everything with the same bit of DNA. And so we scan barcodes by comparing them against reference libraries of barcodes. And we use barcodes for species discovery and identification. And the first paper on barcoding was written by Paul Hébert, who is the person that Robin and I work for. A very nice man. So it's this paper, Biological Identifications Through DNA Barcodes. If you haven't read the paper, you might, it's very short, you might want to. It's very easy to read.
Some suggested homework there.
Suggested homework. No need, but you can.
Probably
And when barcoding was just starting, there was a question of, you want to use the same gene for everything. That's where barcoding is powerful. But which gene you use is very important. And so originally they wanted to use a mitochondrial gene instead of a nuclear gene. And that's for kind of 3 reasons. One is they don't have introns. Does anyone know what an intron is? Yeah, everyone knows? Okay, so sort of coding and non-coding parts of DNA, right? So mitochondrial DNA is all coding for the genes. There's very limited recombination, if any, because there's only one copy, haploid. And so because it's haploid, there's no heterozygosity, right? It's just one sequence of DNA. So if you had a nuclear gene, you'd actually have for every organism, if it was diploid, 2 barcodes, right? Because you have 2 copies of each thing, slightly different. But mitochondria, you have 1. And it's pretty similar between the species. And so in animals, there are 13 genes in the mitochondria. And the gene that was chosen originally was what we call CO1, or cytochrome C oxidase subunit 1. Is that right, Robin?
Yeah.
Okay. We just all say CO1, so I sometimes forget. That was chosen because in the early days, in the early 2000s, 90s, they were making phylogenies. CO1 was the best mitochondrial gene for making a phylogeny, so showing the relationships of different organisms. It had very good primers. Who knows what a primer is?
Primer?
Okay, so a primer is what you... anyone want to explain what a primer is? Who knows? Do you know, Fernanda? It's okay. Manuel?
Primer?
Yeah.
Primer is a short sequence of DNA. This sequence hybridate with the sequence sequence of interest, mitochondrial interest. You can the PCR, for example, for amplification this barcode.
Yes, exactly. So the primer.
I have a talk, but PCR is going to be part of the talk.
Okay.
So go.
Yeah, Robin will explain in more details, but it's the molecule, very small molecule you use to amplify with PCR the molecule, the gene that you want to sequence. And so you need primers to do PCR to amplify the gene, and this gene CO1 had good primers available that worked in many organisms. So that was good, right? You can use the similar primer in vertebrates as in invertebrates, for example. And it has the slowest amino acid divergence of all the mitochondrial genes. So it's fast enough that you can tell apart species, but not so fast that it gets sort of redundant, if that makes sense. And so since barcoding was founded, it's been a big part of the taxonomic community. And we use barcoding to identify new species. And you can sort of see here how many species in the orange line are described with traditional taxonomy, type of taxonomy that many of you, it's very valuable, do. And you can see on the green line how many species identified by barcodes have been identified. And we think in 2 years, there'll be more barcode species than Linnaean named species. But it's a misconception that I think these two approaches are competing. I think they work really well together. And actually at the Center for Biodiversity Genomics where we work, we hire many taxonomists from all over the world to associate barcodes with taxonomy. It's a good way to preserve the knowledge of taxonomists from today into the future, because the morphological IDs are difficult, and if you have metabar coding, you don't have the specimen. So you need the barcode to tell what was in there, or eDNA. It's similar. And this week, one day, who knows, what's his name? Hidalgo Ricardo? Is that, you know him? The Lepidoptera, what's his name? Hidalgo? Anyone know him?
La Peruvia.
Yeah, Lepidoptera, Hidalgo Ricardo?
Lamas.
I don't know. He works on mariposa. Yeah. Ricardo Lamas?
Gerardo.
Hidalgo. Gerardo?
Yeah.
Lamas.
Mhm.
Okay, Gerardo Lamas, sorry. Okay. I'm going to try to convince him at night when he works to let us barcode his mariposa. And so, yes, so in 2 years, it'll be a big, probably celebration when we have more barcode species than traditional taxonomy. But the goal is eventually to have taxonomists work on all these species to associate them. And so, the reason we like to use barcodes is identifying, as some of you probably know, identifying species with morphology takes a very long time. You have to, sometimes, like what I brought some cycodid flies to Mayville from Guelph, and that involved me putting them on a plane and carrying them here and giving them to her. But once she barcodes, we barcode the ones that she identifies, or we've already barcoded them. Once she tells us what species they are, then to identify the species, all people need to do is barcode. So we can, the taxonomists can work on the difficult things, and the routine things we can use barcoding for. And DNA barcoding works very well for most insects and other orders. More than 95% of species you can identify accurately with barcodes. Some groups, it's more difficult. Yeah, it's a highly viable approach. Different genes are used sometimes for different organisms. In animals, almost everything is CO1. Some people use 12S, 16S, or cytochrome B. Birds often cytochrome B. Plants are more difficult. People use nuclear genes and chloroplast genes. Right now, I think the main ones are ITS and RBCL people use. Fungi, people use ITS, 18S mostly. Bacteria, 16S. And protists, people use different genes as well. And the gene is usually chosen by communities working in the groups, and they find what works the best for the group. And they all have their advantages and disadvantages. But one thing is they have to be not too long, because PCR works well for short molecules, but it's harder as the molecules get bigger. So I'll just very, very briefly go over this because Robin will talk more. So the way you do barcoding, and we'll do almost all of this: sample tissue, extract DNA, amplify the DNA with PCR, make more copies of it. That's what that does. Sequence the amplicons, we call them, amplified products. Takes the data, put it into barcodes, the DNA sequence that's all lined up, and then you make a tree and you compare sequences. That's the broad workflow. Robin will go into many more details.
Ken,
Yep.
Ken, it is true that we need to use more markers. Is better for the accuracy, the identification, or no?
So the question is having more markers, so say 2, better than 1? Sometimes, most of the time, no. So the DNA barcode is, and we can do some exercises on this with the class, actually, more advanced exercise. Let's hope this works. The DNA barcode that we use, CO1, is 658 base pairs. And I find in my own research, actually, if you only have a piece like this, for most species, that's just as good as the whole thing. So I find that. And I think one of the exercises we can do at the end of the project is to have this more advanced, have you go through different species that are on the internet and see how well small parts of the barcode work to get the right species. So that's one thing we can do. Some groups, multiple genes work better, but
kind of situation specific.
Yeah.
Like one example could be where you have a genus of very closely related species, a recently diverged genus, and maybe call1, maybe there hasn't been enough time for variation to kind of accumulate in call1. Maybe call1 is identical between those, but we really do think they're different species because of morphology. So in that case, you might pick a different gene, a faster evolving gene that might have differences between the species and call1 doesn't. But that's it's kind of like 95% of the time, the call1 algorithm will give you enough information to discriminate species. It's more just odd situations that are maybe different for one reason or another. You have to go to a different one. So yeah, kind of situation specific.
Yeah. So in my PhD, in my PhD, I work on these fish, and they hybridize all the time. And so the mitochondria of one goes to the other one all the time. And so in that case, barcoding doesn't work well. But actually, no gene works well. You need to use the whole genome. And so what we would do is we would sequence, use an approach called RAD-seq. There's dd-RAD-seq, restricted. So you sequence 150 base pair segments very spread out on the genome, and you use those to determine which species you have. So it's more blurry. But for well-established species, barcoding works quite well. Yeah.
Or does it depend on the variable region of the gene we chose?
Does it depend on which region, sorry?
Of the gene chosen.
Does it depend on the gene chosen?
For example, for bacteria, with B3, B4.
I don't know. Do you know about bacteria B3, B4?
I don't know about bacteria. I think they have, people have mostly used bacteria. 16S, I think people have done quite a lot of work with 16S for bacteria, and it's, I guess it's usually the same region, but maybe you choose different regions depending
Some
on the
words
bacteria.
use B1, some words B3, B4.
These are maybe versions where they've got different amplicons of the gene.
Yeah,
so
probably. I don't know a lot about the specifics of that, but yeah, bacteria are probably more complicated.
The better your reference library. So the more species you have and the more individuals of the different species, then it'll work better. But some genes will just, by their nature, work better. So if you use a very fast evolving gene, the differences between the species can become redundant. So yeah, it depends a lot on the gene. And that's why usually it's communities decide on which one to do. So it's a balance. Yeah.
In the case of plants, reporting in plants, I read in a lot of investigation papers use two workers.
Yeah.
Two markers are often used for
Yes.
plants, yeah.
It's the same in all the investigation of reporting in plants.
It's similar. So people use generally, I think now, RBCL is common and ITS. Is that what you would say, or MACK sometimes?
Yes, and
MACK, yeah. So I'm not an expert in plants. I don't know, Robert, maybe you.
Yeah, I don't know a lot about plants either. I think I think between RBCL and ITS, I think RBCL is the less variable one, so it can kind of put a plant to like the larger scale plant groups, but it will tend to be the same between closely related species, and ITS will be maybe different between the closely related species, but it's too variable to place it to higher groups if it's far from anything else. So it's kind of like you just need the two different sources of information really. One will give you, one is better for like higher level taxonomy for larger groups, and one is better for species level taxonomy.
Yeah.
But
So
obviously you have to have reference libraries for both of those things to get a map. So.
at every spot in the genome, there are only 4 possibilities, right? A, T, C, G. And when you have a little bit of evolution, you might go from A to T and then maybe T to C. But then if you have a lot of evolution, you'll go back maybe to A. And so what happens is when things evolve a lot, if the gene evolves very fast, it actually erases the signal, because you develop a lot of signal, but then things just keep evolving, and it becomes very difficult to tell the evolutionary history. So with fast evolving genes, there's less signal phylogenetically. With slow evolving genes, it takes a long time to get that. And so you can use a slow evolving gene so the phylogenetic signal is maintained over deep time. I think that's what RBCL is used for. So a slow evolving gene, you can tell maybe what family or what genus you're in. But some genes are so fast that you can't do that. You could have the same, very similar sequence in, maples as in oaks or something, because it's too fast. But you can use like MAC-K or ITS, which evolves really fast, to tell apart the closely related species. So you can use RBCL maybe to say, okay, this is in Jugulandaceae or something, and then the other one to pick the species. Does that make sense? So it's a two-tiered approach. Yeah.
But from what I remember about ITS, looking at some of Andreas's data, it's so variable that you can't even align it a lot of the time in different plant groups. So it's you need to already know what genus or group of species you're in for ITS really to be useful.
Yeah. So slow evolving gene to pick the small taxonomy level, genus, family maybe, genus, and then fast to tell within. So that's more how it works. Yeah.
Yes, I have a question about that discussion. So, in order to this question, so if we use more than one gene, like coding, so we can talk about DNA barcoding, because it's supposed to make barcoding, just use a standardized gene. So maybe we can talk for example, in the food species delimitation, if we use more than one gene, or it's the same, I don't know.
I think
this question.
so. The question is, if I understand, if you use multiple genes, are you still doing barcoding? Kind of. Yeah. I would say barcoding is just the approach that uses a small number of genes, but you can use 3 or 4, and uses the gene sequence of the whole gene with a reference library to compare. That's what barcoding is. So you have the reference library and you have a small number of genes with references. If you're going to sort of like what I did in my PhD and postdoc, we're doing whole genome sequencing, then that's not barcoding. But I think if you're comparing a small number of gene sequences against the reference library,
yeah, it needs to be a particular subset of markers, and that'll usually mean CO1. In animals, certainly 90% of the time, if someone says DNA barcoding, they're talking about CO1. But it can potentially be other markers too.
More than one gene. For example, for bacteria, he said that use two markers, right?
I think people tend to talk about molecular taxonomy in bacteria rather than molecular barcoding. Would you say that's right? Yeah. I mean, that's kind of been a standard approach in bacteria for longer than it has in plants and animals, because, well, for a lot of bacteria, that is the only way you can get information about the sequences.
Sorry, my point is not that if we can change the markers, is if we use more than one markers, if we still name DNA barcoding.
Yes.
Yes, if they are the limited subset of markers, and I think that the Angol's website, or there's some website that has these are the set of markers and bold, it will say these are the set of markers that are considered DNA barcoding by the community. You can use other markers as well in your own research, but that's just general molecular taxonomy rather than molecular barcoding, specific thing. Okay.
Yeah.
I just want to make a comment about a question previously that was about the choice of viral regions for the markers for bacteria. And this choice is generally with short reads
So this was related to Fernando's question. The V1, V2, V3, V4 was because you can only sequence small parts. But now that we have nanopore, PacBio, we sequence the bigger ones, the choice of the subset doesn't matter as much.
Yeah, that sort of goes to the sequencing technology available. The 658 base pairs really links to Sanger sequencing because that's about the maximum amount of data you can get in a bidirectional Sanger read. And now, of course, nowadays, if we'd been using Oxford Nanopore all along, which has an almost unlimited read length, maybe we would have picked a longer marker. This is not the full cytochrome oxidase, by the way.
No,
it's only about half of the actual cytochrome oxidase gene, which is about 1,500 base pairs long. But this was sort of established, and now it's sort of become the standard because there's so much data. There's so many species and organisms for that 658 base pairs. We just keep using that, even though potentially we could use longer markers now.
But also, as I was saying, adding more doesn't necessarily, you don't learn more.
Yeah.
Like adding more.
Well, that's true. I mean, the other reason is it works. You know, this does actually tell apart most species, so you don't actually need to get more information one at a time.
Yeah.
And in fact, for eDNA, I mean, another thing to consider is that depending on the source, eDNA is often very fragmented. That's environmental DNA, DNA in water or air. It's often short. You can't amplify 658 base pairs because all of the DNA there is chopped up. It's just little bits. So you have to go to shorter markers in that situation.
Okay, any more questions? Okay. So let's go into some methods and sampling. So at where we work, the Biodiversity of Genomics, the largest team works on this part, which is the sample processing. We call them the collections and imaging team. So samples come in, ideally they're in ethanol or 20 degrees, so minus 20 C freezing. But dried and pinned is okay. The DNA just degrades fast. But one of you was saying you work with degraded DNA, in eDNA. It's important because we want to sequence the whole molecule that the DNA is intact. So keeping the DNA fresh is important. For small things, we sequence the whole thing, the whole animal. We just put it in a small well in the DNA plate, in a 6-well plate. And for big things, we take a piece of the leg, usually. And then we take a photo of each one. And this is a photo of what's called our I think it's called our Keon's machine at CBG. And that you can see it's the microscope, and it's zooming in on a well in a plate, and that's a specimen in ethanol. And it takes a photo of each one, and it moves the
Automated.
It's automated. Yeah. So it takes a photo of each well, and then saves the files in an order so we know which file is which insect. And so we take the photo, put everything in a database, and we keep everything. So we have a big museum. Unless people want them back, we keep everything. So all the DNA, we've got many, many freezers, and we have many, many drawers and cabinets like a museum of pinned specimens. DNA extraction, Robin will talk a little bit more about this, right? So I can just zoom through it.
Let's get through this.
Yeah, you have to get the tissue, turn it into DNA so you can use your PCR. Robin will talk more. PCR, Robin, you'll talk more about this in your new lecture. So essentially what you do is you make PCR polymerase chain reaction. You make many copies of your DNA so you can sequence it many times. And then Robin will talk more about sequencing, but you take the amplicons that you make from PCR that you prepare for the sequencer, put them into a machine. These are the two main, this is a slightly older version, but two main nanopore instruments. The big one, anyone know what that's called? See it? The small one is yeah, so the little one. We have them. Maybe Robin's pulling it up now. Small one is minion. Big one is promethion. So those are the two sequencers that nanopore makes, so that's one right there. So it's really small.
You can come up and take a look after that if you want to see it.
That's the Mark 1B.
Opens up, you put yourself. Yeah, the Mark 1B, so the older one.
Did we bring the Mark 1B?
We did not bring the Mark 1B.
And then so you use the minion. It's cheaper, and you use that for small. And you can use that for DNA barcoding you don't need to use a Promethean. Minion is fine. We used the Promethean once for DNA barcoding, Robin did, and our colleagues to sequence 1 million specimens, which is crazy, at one time. And then we did, 100,000 on the Minion you did, Robin?
Yeah, you can do it.
But for almost anybody, those little tiny sequencers for barcoding, that's enough. That's all you need. You don't need to spend more money.
We've taken these to Los Amigos and done this at field stations.
Yeah. So actually, as a part of this course, you will analyze data that Robert and I generated on a Minion in Peru in April, which is fun. And so we brought it from Canada and did all the sequencing in a field station in Los Amigos and Madre de Dios. And for people who are doing eDNA or metabarcoding runs with many samples at the same time, then you might want a Promethean. But those are more expensive, but more expensive for the machine, but cheaper for the data. So yeah. Skip this. So once we have the DNA sequence, it's just A's, T's, C's, and G's. So you need to use a computer algorithm to tell, you could look by eye, but it would take forever. You use a computer algorithm to match the sequence to the reference library. So essentially what you do is you have one sequence. So this is like maybe your focal sequence. And then you have your reference library on bold. So this is reference. And you have million sequences. So you need to check each one against each one. And so if you did it yourself, it would take forever. So people have very smart, it's like when you search on Google, you search on Google, it's fast because people have written algorithms to really reduce the amount of information that's being checked quickly. It's the same thing here. So people have written different algorithms, bold at the engine or blast, which is common, syntax. We use, in this course, we'll use vsearch, it's called Protax, to do this querying. So you would never, it's really high-level computer scientists that develop these approaches. You would never write a program to do that yourself. And then once you have a match, you'll get a match 99%, 75%. Once you have a match, you can use the information in the matches to tell you about what it is. If you notice, for example, that you have 99% match to one species, 99% match to another species, then maybe that's just one species, right? So you can sort of use that. And even if you don't get a match, let's say, you had, for this one you had 96%, and then this one you had 95%. So you might say, that's close, but for a match, we usually say 2%, 2.2% is what we see within species. What we could say is that's probably in the same genus, right? Based on that. Or if it's two things with 90%, you might say, okay, it's probably the same family or subfamily. So even if you don't have something in your reference library that's an exact match, you can still get a lot of information. And when we worked in Peru in April, 1 in 3 species, we didn't get a match. But we could tell what family they were. So many new species, that's how you can sort of tell. And so then I say, I look at this bug, Hemiptera, and I say, okay, I know it's a Hemiptera, but I get the family ID. And so I say, okay, I know this guy is an expert in this family, right? So if I had an Acylidae, I would send it to you. But if it's an amateur, they might not look at it and know it's an Acylidae. But the barcode, you can tell. And it's also really nice because sometimes if you have an ID based on the adult, the larva is just a little worm, right? So you can never ID that, right? Like, do you ever work on larvae?
No.
No, right? Do you work on larvae? You work on larval? No, right? So the taxonomic experts, they don't work on larvae, right? So your morphology IDs.
Even females in my case are difficult to identify.
Even females are difficult, exactly. So different sexes, right? Dimorphic sexes. So if you have an expert, maybe working in males and adults. If you find a female or if you find a larvae, the barcode is the same. So you can identify different life stages, right? So it's very useful for that as well. Or also if you only have an antenna or a leg, you want to know what it was. Like a mosquito bit you, but I smacked it and I only have the leg. What is it? You can tell, right? So that's what it can be very useful for. So within species, we see there's a lot of cryptic species, very similar. The cryptic species they're morphologically highly similar, but genetically very distinct. I don't know if any of you deal with these. It's very common, cryptic species. Barcoding can tell those apart too. And what we started doing sometimes is we can use barcodes to be very confident that we have a new species, but if we don't have an expert, but we want to still describe the species, we can just use barcodes and limited taxonomic information to do it, which is nice. And recently, me and a couple colleagues and Paul described a new species of moth in a family that only had one species. So now we added a second one. And so we could use barcodes to be confident we had a new species and then just say what we found. But the main point is sequences within a species are close. Sequences between species are usually far. And that's how barcoding can work. And so the new species we found of moth in Tridentiformidae, minimum 7% difference from the other species. So we were confident it was different. You can also use barcoding to study species interactions. So sometimes maybe you sequence a fly and you get a fly sequence, but you also get a wasp, right? And that could be the larvae of a parasitoid wasp in the fly. So you would call those a non-target sequence, right? So you can use those, you get multiple, or bacteria, for example, in the gut. You can use these non-target sequences to identify the relationships between different species. We see this pretty often. People also use barcoding for conservation. You know, someone says maybe it's illegal to do poaching, for example, of a certain species. Like I think in Peru, there's like llama fur. You can take llama fur, llama, but not the native one. What's it called? The brown one?
The llama?
The wild llama.
Llama.
Picuña. Picuña? See, so picuña. So if somebody is brown, right?
Yes.
So someone takes a picuña fur, they dye it a different color, or they can say, no, it's llama, it's llama. But you can do DNA barcoding on the fur or the skin and tell, no, it's picuña, it's not llama, right? So you can use barcoding to distinguish this, right? Or you go to Maido, fancy restaurant, Maido in Miraflores, and they say they're giving you a very fancy fish. You take a piece of it, and you say, that's not the fancy fish, that's, you can, there's a good restaurant, seems like a good restaurant. Too expensive for you. So food mislabeling, people use this for. And also, I think increasingly what we will do is using our malaise traps, which are insect traps, you can detect species that are invasive, right? So you say, we see this species all the time in Europe, in our traps. We've had a lot of traps in North America, we've never seen this. But if it starts showing up, then we can say, okay, we have a new invasive species, it's in this area, right? And you can do the same in eDNA in the water, for example. So you can detect invasive species. So that's often applications what people are doing. Barcoding also is used a lot for teaching and community engagement. So one thing that our organization has done is we work with schools, set up traps for DNA barcoding. And so the students go out and they collect the samples, they set up the traps, and they learn about the biodiversity in their area by doing this. And we also then use that to build the global reference library. And so increasingly, I think also with our software that we've made, a lot of people can do this type of work to learn about biodiversity. So we talked about a bit, barcoding is not perfect. There are some cases where you wouldn't want to use it. So for my research in speciation in the past, I wouldn't want to use barcoding because the species are hybridizing too much, right? You need to use whole genome approaches. There's not a, we have these two fish species. There's not a single gene, there's not a single part of the genome that is unique to one. It's all, you know, probability, right? Chances are you have this or this species. So in some cases, so you can have a lot of this hybridization and non-exclusive barcodes with certain groups that make barcoding less useful. Sometimes, like in some wasp species, barcoding can be difficult. The primers that we use, for example, don't work as well on those wasps. And so for some taxa, the general approaches are challenging. And also you can see things like there's parts of the mitochondrial genome, which comes from a bacteria originally, that are in the nucleus, but they're not CO1, they're a version of CO1. And so you can get these, we call them pneumites or nuclear mitochondrial DNA segments.
Pseudogenes.
Pseudogenes.
Is the proper name for that type of thing, a non-functioning copy of a gene.
And that can, so you can have something that looks like CO1 but is not. So you have to, there's some challenges with recognizing those. So some groups like grasshoppers or Orthoptera have a lot of pseudogenes. Yeah, and there's all sorts of chance you can have contamination. So for example, you can have something that you think is one species, but you maybe it ate, like if a Acyllidae or something, it eats other insects, right? You can have, you can get one sequence out, but maybe it's not the Acyllidae, maybe it's something else. And then you can have a bad sequence. So you can have some, if you're not careful. Of course, any molecular process, you can have contamination in the lab, that can be challenging. And yeah, some taxa amplify better than others. So if you have a wasp that's been parasitized by something else maybe, you might get the parasite over the wasp because wasps can be difficult. And then the last thing is barcoding, kind of because you use reference libraries, it's only as good as the reference library. So we have at CBG, working with our international communities, we have barcoded about 1.3 million species so far, which is good. But there's probably 10 to 20 million species on Earth. So many times we get DNA molecules that we don't know what they are. And so, especially in regions like, for example, in Madre de Dios in Peru, Iquitos in Peru, many people haven't been there to do barcoding, so there's very little known. So if you do barcoding in Iquitos, you won't know what you're working with because no one has barcoded those, for example, or in groups that people haven't really worked on, geographic groups. And it's also like in Peru, it's difficult to export. So part of the reason we don't have a lot of data from Peru is because it's very difficult to get export permits for us to take the animals from Peru back to Canada to do the science with them. The government doesn't really, it's not so easy to do that. So we have to come here to do it. And if there's not the sequencing facilities, you can't do barcoding in Peru. But we're trying to change that now with our technology that we're developing. Okay, so just to wrap up this, DNA barcoding, you can get high throughput, fast, accurate species identification and identifying new species. It's used very often in primary research, biological research, conservation, veterinary applications, other applications, but it's only as good as the reference library. So that's really the big mission of the organization, IBOL, CBG, is to build the reference library. And we work with taxonomists to do that. So, yeah, any questions about this? That's the last for this introduction lecture. Yeah, honey.
I was thinking about the pseudogenes.
Pseudogenes?
Yes,
Yep.
and it may be a bigger problem when we work with metabar because we lose extra.
You're correct. It can
A
be,
high
yes.
number of yes,
Yeah.
a high number of access that we have in our environmental sample.
Yeah. Yeah, Robert, do you want to say something
Yeah,
about that?
totally correct. When you're doing metabar coding or using which is kind of metabar coding, you're dealing with a whole mixed set of DNA, and you're just amplifying whatever will amplify in that set. And yes, that can include pseudogenes. If you have a pseudogene version of COL1, and if it still has primer sites that are similar enough to the real COL1, then yes, you'll amplify that as well. Pseudogenes, of course, because they're non-functional, like the longer they stay in a genome, the more they kind of accumulate differences, and they become like different. Eventually, primer sites will not exist because there's no selective pressure on them, right? Because they're not.
They drift.
They're genetic drift. They're not functional genes, so there's no selection keeping their sequence the same. So. Yes, you need ways of recognizing pseudogenes when you do metabar coding because you will amplify them. Usually they can be recognized because they have kind of telltale differences from a real functional COL1. If you translate the sequence, they will often have stop codons in them. You know, because they accumulate changes that are random and they don't have any effect on gene function because pseudogenes don't have any gene function. You can have a randomly created stop codon, for example. Say, okay, if this has a stop codon in it, it definitely can't be a real functional COL1. So just write that off. That's a pseudogene. You can have length variations. The barcode region is very consistently 658. It can vary a bit, but it will always be multiples of 3. You know, because of the 3-base coding frame. So if it has a length that's outside of that, then you either know it's either got sequencing errors or you've got insertions or deletions. And that pretty much means it has to be pseudogene because you don't get insertions or deletions, real COL1. Sometimes you see things, sometimes I get sequences that's a wildly wrong length. That's like 100 base pairs different. And if you align it to a real COL1 sequence, you can see it's either missing a huge chunk or it's got a huge chunk of DNA that's not in a normal COL1. So again, those you can immediately tell. Those are pseudogenes, so you just ignore those. You're not going to get sensible matches to those in the reference line. So yeah, there are ways to tell, but also those are the obvious cases. There are cases that are not obvious, where you have pseudogenes that are just maybe very recently became pseudogenes, so they haven't had time to accumulate these kind of changes. And yes, there probably are instances of things where there's pseudogenes that we can't recognize, maybe have even been incorporated into reference libraries like gold that we haven't recognized. It actually doesn't matter too much in that sense because it still effectively is a marker for that species. You know, that is a gene that comes in that species, even if it's not COL1. So yeah, I mean, it is an issue, and there are ways of recognizing when you have pseudogenes, which I think we'll talk about a bit more when we do the bioinformatics stuff. But yes, you're right to mention that as an issue.
Yeah. Yeah.
In a research project, what is the average percentage of unrecognized animals?
So the question is, what is the average percent that's unrecognized? It depends primarily on where you're working and what species you're working on. So if you're working on fish in Canada, 0. Because pretty much, we have done them all. Birds in Canada, 0, probably. But insects in Canada, some. Insects in Peru, a lot. So it depends primarily on that, where you're working. So the more people who have worked in that group and in that area, the more you'll know. Yeah.
And it depends on, I mean, the more obscure groups. My PhD was on nematode worms, and we still do some work on COL1 and nematodes. And if you an average set of nematode worms from like soil or something, you will often get no exact matches to a known species, just because there are so many species out there, and so there's so few people studying them that you'll often not get an exact match, but you might get a good enough match to place it to a family or whatever. You know, we did some work on mites and collembolans, tiny things that live in soil, and this was in Canada, so this was in local stuff, but we got exact matches for almost nothing. So, because again, there's so many species and they're so hard to identify, you'll maybe get, okay, it's close enough to say it's probably this genus or it's probably this family, but in that situation you don't get exact matches. But that can still be useful information to have, general families.
Yeah, so if you're working in an area people haven't worked, or in a group of organisms that people don't work often, that's challenging, a few matches. Yeah. In Peru, we sequenced 1,000, mostly butterflies, roughly, and we got match for about 2/3. So 600 or so sequences. And then of the species, yeah, 2/3. Yeah. Any more questions? Okay, let's take 10-minute.
Yeah, good time for a break.
Yeah, let's so at 11, we'll come back and we'll do the first program.
Okay.
Okay.